Ladies and gentlemen, welcome back after the break. Now we will be moving on to lecture number 3, which will be delivered by Dr. Normala Latif from Faculty of Pharmacy, UITM, Puncak Alam Campus, Bandar Puncak Alam, Selangor, Malaysia, with a lecture entitled PFAS, Exposure and Metabolic Diseases. This lecture will be moderated by Apothekar Medi Utama Putri, MSc PhD from the Faculty of Pharmacy, Universitas Indonesia. Before we jump into the lecture, please allow me to read Dr. Medi Utami Putri's CV. Dr. Medi Utami Putri, MSc PhD, completed her Bachelor of Pharmacy and Professional Pharmacist degrees at the Faculty of Pharmacy, Universitas Indonesia in 2013 and 2014. She pursued her Master of Science in Medical Sciences 2014 and PhD in Biomedical Sciences 2016 at the University of Tsukuba, Japan, funded by the MEXT Scholarship. Afterward, she worked as a postdoctoral researcher specializing in molecular cancer biology, focusing on the roles of TMEPAI in cancer stemness induction. Currently, she is a lecturer and researcher at FFUI in pharmacology and toxicology with research on tumorigenesis and related diseases. All right, without further ado, please welcome our honorable moderator, Dr. Mehdi. The time and floor is yours.
Okay, thank you, MC, for the nice introduction. So, good afternoon, everyone. My name is Midi, and I will be your moderator for today's lecture entitled PFAS, Exposure and Metabolic Diseases, that will be presented by Dr. Nurmala Abdul Latif from Faculty of Pharmacy UITM, Puncak Alam, Selangor, Malaysia. So, welcome, Dr. Mala. Thank you for accept our invitation. It's our pleasure to have you. So before we begin the lecture, please allow me to read Dr. Normala's CV. So Dr. Normala is a research fellow in the Integrative Pharmacogenomics Institute, I promise, University Technology Mara, UITM, Malaysia. Her research focuses on pharmacology, particularly the genetic variations of the metabolic enzymes and pharmacokinetics. Her projects primarily investigate the interaction between pharmaceutical compounds and xenobiotics, along with their combined effects on cells. She frequently utilizes cell culture models, gene modification techniques, and advanced chromatographic methods to measure metabolite concentration in biological samples, which is essential for understanding pharmacodynamics and pharmacokinetics. Currently, her research interest lies in exploring the effects of environmental pollutants on metabolic enzymes. So, all right, without further ado, please welcome to our honorable lecturer, Dr. Normala. To Dr. Normala, the time and screen are yours.
Assalamualaikum. Can you hear me?
Yeah, Waalaikumsalam, Dokter.
Okay, good afternoon, everyone. Thank you for a very generous introduction from Dr. Mehdi. And first of all, thank you for the invitation for this lecture to share my humble research topic that currently is still ongoing. And let me share my slide first, I think. Okay, previously I gave the topic on PFAS and metabolic syndrome, but with the interest of time, I think I want to share just one small topic of the research with you. It's one of the PFAS, perfluoroalkyl substances, which is PFAS, the perfluorooctane sulfonic acid, and its correlation with cardiovascular diseases. So this is very close to my heart because it's a major concern in Malaysia. I will show you why in the Next slides, I think. In Malaysia, we have this problem, obesity, metabolic syndrome, basically non-communicable disease. I think in Southeast Asia, we are really blessed. Indonesia, Malaysia, Brunei, Thailand, Vietnam, we are really blessed with many foods, various types of herbs and, you know, recipes. We are very creative as well to market our food, and we are really enjoying those food without much of restriction. And currently, it's a major concern, especially in Malaysia. So cardiovascular disease, for instance, is a leading cause of death in Malaysia. It makes up around 15% and sometimes goes up to 23% medically certified death each year. So this basically the example of the report each year that we receive from the government. In 2020, for example, we have 17% cause of death by IHD or IHD, ischemic heart diseases, and in 2023, it's around 16%. So the numbers is not going down, it's between 15 to 25% each year. So, and another thing is 1 in 5 adults in Malaysia have diabetes, 3 in 10 person or adult in Malaysia have hypertension, and 4 in 10 is actually having or currently being treated with hyperlipidemia. So these are the contributing factors of cardiovascular diseases, and it is on the rise in Malaysia. And Malaysia is currently juara Southeast Asia in terms of obesity. I think we miss the second champion, which is Brunei. Indonesia is still doing good, not so bad, but Malaysia is bad. We have the endemic of obesity, especially in children. And I think if this is not curbed earlier, we will have major problem in the future as well. So this is the comparison of cardiovascular disease in Asia. And as you can see at the bottom right, Malaysia and Indonesia is quite similar in terms of percentage of prevalence of cardiovascular disease in Southeast Asia. Okay, so many people have looked into the risk or biological attributes in cardiovascular disease in terms of biological attributes. Those are, and we have non-biological attributes like physical activity, the lifestyle, tobacco smoking, and another one is environmental. So my group interest is on the environmental pollutant. Previously, we got a few grants to study the environmental pollutant in our water sources and the correlation of the or the health impact in our population. What is PFAS? It's actually we can say it is a forever chemical, and unlike BPA or the common thing like PCB, people are aware of the danger of those compounds. PFAS is kind of a newcomer. It's actually a plasticizer. It's being used a lot in our daily life, but we don't really kind of aware about it. I will share on it in a few slides, I think. Yeah. So this is a growing concern because PFAS, the oldest PFAS, probably the PFAS and PFOA, the perfluorooctanoic acid, the acidic part of the members. But we have a few other generations of plasticizers that we don't know of. And as of now, currently in Malaysia, we don't have any regulation on the level of safety, or regulation on whether you can pollute the river on with this compound, in which level that we can curb. Or even if people pollute our water resources with PFAS, we don't have any regulation to do anything to them. So yeah, it's a carbon compound. Basically, it's a non-biodegradable. It's a forever chemical and highly lipophilic, so it's easy for us human to be distributed in our system. So it's easy for us to absorb this compound and to retain it in our tissue. Yeah, it is persistent and bioaccumulate. We try to see the impact on phytoplankton and the fish as well, and it's there. So it's bioaccumulate as we go to the higher order of animal. So the end user is us human. So we get the brunt of the end of environmental impact, and it's really bad for our health that we know of nowadays. We know the danger of this compound, but it's not really studied on how the impact in human being precisely. And the sources of PFAS, basically industrial sources, wastewater, firefighting foam. This is not common, but the most common one is the consumer product, which is the non-stick cookware, the plastic or food packaging, textile, and water bottle, and so on. So I don't know about Indonesian, but in Malaysia, mothers basically, or grandmothers, they don't want to throw away the plastic container. It's like they are holding the plastic container and using it over and over again, the cheap plastic container, and it can actually leach into your food as you keep the food inside the container, and it will affect your system, gets into your body system, and give impact to your health. Okay, so the potential mechanism on PFAS, of PFAS on cardiovascular disease, basically, it is called EDC, endocrine disruptors compound, disrupting compound, or endocrine disruptors. It can cause oxidative stress to our system, and particularly it can cause inflammation in our system that leads to many other diseases such as cancer, diabetes, and cardiovascular disease. So first, we want to study the PFAS and its impact to cardiovascular disease. But which type of PFAS? Because we have hundreds of PFAS, and the numbers, the types of PFAS is growing. So first, we did this study, the meta-analysis study, to sort of like reduce down the most significant one, because if we want to see the most significant one that impacting our local population. We found that PFOA and PFOS exposure were found to be associated with cardiovascular disease, and so we focus on PFOS and PFOA. But in this today, in this discussion, we will only discuss on PFOS. So this is PFAS. It's a carbon fluorinated compound with hydrophobic tail and slightly hydrophilic head. So it is kind of easy to be distributed in water, and as we consume it, it is easy to be absorbed in our system. Okay, so this is a study done in 2012. We can see that we already have contamination of PFOS and PFOA in our water sources in Malaysia since 2012. And this is the study in Langat River Basin, the major water sources for people in Kuala Lumpur and Selangor, and the most populated area in Malaysia. The impact on drinking water is not bad in Malaysia, but in comparison with other countries such as China, but it is a growing concern, and I think it's worth to study this compound. So first, we want to we sort of like recruit the patient in our primary clinic to give blood so that we can measure the amount of PFOS in their blood, and we try to correlate the level of PFOS with the condition that they already have. Basically, this patient is coming to the clinic for regular checkup for hypertension. So we have a control group, kind of healthy people, and hypertensive group. So we did the LCMS profiling on their serum, and we can see PFOS is available in 10% of the patient that has hypertensive, especially positively correlated with elevated diastolic and systolic blood pressure. PFOS as well, available in 25% of the hypertensive patient, with positive correlation of higher waist circumference and higher weight as well. In terms of lipid profile and sugar level, as you can see, PFOS is you can see that PFOS is actually elevated, kind of, in the hypertensive patient. Yep, in the hypertensive patient, and it is correlated with the higher level of cholesterol and higher or elevated fasting blood sugar. So as a conclusion, we can see the increased risk of 10% of hypertensive patient with regards of PFOS concentration, and PFOS in hypertensive versus I have kind of typos there. In normal versus hypertensive, betul lah. In normal versus hypertensive is associated with high triglyceride, LDL, and lower LDL, and it is correlated with higher fasting blood sugar and hemoglobin, HbA1c, and higher waist circumference and weight. So all the attributes that we discovered is actually correlated with the metabolic syndrome. So these patients are at higher risk to be diagnosed with, metabolic syndrome. And metabolic syndrome is one of the major cardiovascular diseases one of the major consequences of metabolic syndrome. So as you can see, this is the sort of like progression of metabolic syndrome to cardiovascular disease. You would have elevated elevated lipid level, insulin resistant or diabetes, metabolic syndrome, and eventually you would have full-blown type 2 diabetes and cardiovascular disease. So all the attributes with correlation with PFAS actually increase the risk of the patient, hypertensive patient, to get cardiovascular disease. So so that's done for surveillance or for to see whether PFAS is available in the serum of patient with correlation of metabolic syndrome, and further on the elevated risk of cardiovascular disease. So next we want to see how PFAS might have or might contribute to cardiovascular disease later on. So what are the possible mechanisms that PFAS exposure can induce cardiovascular disease? So we want to see the effect of PFAS exposure directly to the cardiovascular disease. Okay. So we have the in vitro model that will help us to study the mechanism of action of PFAS exposure. In this discussion, I will share on neonate rat cardiomyocyte. So this cell is actually isolated from the babies of rats, and we are using it in our study, in vitro study. So the recently born neonates of rats is isolated from the mother, and we extracted the heart tissue, and we grow the heart tissue on the petri dish. So you can see very beautiful monolayer of cardiomyocytes. And when we have the beautiful monolayer of cell, we can give quite a bit of short electrical shock so that the cell will beat like your heart muscle is beating. So we can actually measure the beating rate of this cell on the petri dish. As you can see, after PFAS exposure, this is compared to control. The higher the exposure, the higher the beating rate. And when we compare the beating rate with normal and positive control, endothelin 1, you can see the PFAS exposure or exposed cell, it has higher beating rate, almost equivalent like the positive control endothelin 1. Then we want to see one of the markers of stress in cardiac muscle, BNP. BNP is being used currently as the biomarker clinically in the clinic to measure or as a biomarker for cardiac injury. So we can see when we compare with control, after the exposure of PFAS, we have elevated of BNP. So we can conclude that the previous observation is not biased. We have very stressed cardiac cells, and it will actually sort of like as an indicator that this cell is prone to have cardiomyopathy. Okay. So yeah, this is the pathway involved. When you have myocardial stretch or stress myocardial tissue, your cell muscle will produce BNP actually to relieve the stress of the heart, if the heart is in our system. So BNP is one of the markers for cardiac injury. So we had then we did the pathway analysis, and we can see a few markers or indicators for pathways that involve in the hypertrophic cardiomyopathy and hypertrophy, and the upstream regulators of hypertrophy and cardiomyopathy in terms of microRNA is available or observed as well. So these are the example of microRNA involved in a few major events in cardiomyocytes. We choose a few that are kind of very relevant to our topic, the hypertrophy, cardiomyopathy, and myocardial infarction. So after the exposure, we can see the elevated level of microRNA 199A3P and a few others. But for the PFAS exposure, for the PFAS exposure, we only can get one positive correlation with microRNA 199A3P. So this particular microRNA is associated directly with hypertrophy and myocardial infarction. Then we measure the protein level as well after the exposure of PFOS and endothelin-1 to see the protein that are important or markers for inflammation to correlate the condition with hypertrophy and cardiac cell injury. So we can see here the elevated level of adenopectin, interleukin 6, leptin, and TNF alpha. So as a conclusion, I think for now, we can sort of like have quite a bit of story that correlates the PFOS exposure with cardiomyopathy or hypertrophy via microRNA 199A3P alteration or expression level. So PFOS is able to sort of like modulate this microRNA 199A3P to drive this cardiac cell to become quiescent and hypertrophy. Okay, so what's next? We got one pathway that directly sort of like tie up PFOS exposure with cardiovascular disease. Next, we are quite ambitious to work with not only PFOS, but we add the PFOA and the legacy of PFAS, the medium chain of PFAS, and we did the in silico screening and network pharmacology to find the candidates and to find the protein-protein interaction and to find the pathway, related pathway that make the interaction between the compounds and its exposure to correlate with metabolic disease. And we are actually genotyping the patient so that we can answer why only 10% of the patient has elevated risk to get cardiovascular disease and not the rest of the 90%. So apart from that, we have this previous work as well on the model cell model of cytochrome P450 and UGTs, as well as SOLT1A1. So we are able to basically do the screening, in vitro screening of the interaction between the compound using this cell model as well, and doing the enzyme assay to see whether the combination of PFOS, a PFAS, component of PFAS will inhibit the metabolic enzyme, or because currently our work is on the single compound, PFOS, PFOA, and probably BPA and PCB. But now we want to see a few compounds together in one system, and whether this interaction can have further impact with towards our metabolic enzyme, and furthermore on how we excrete the compound from our body. Okay, so these are my team members, and we work closely with our teaching hospital, Hospital Al Sultan Abdullah, here in front of the campus, and the Institute of Pathology and Perubatan Makmal and Forensic in UITM as well. So thank you so much. That's my sharing for today.
Thank you, Dr. Mala, for your impressive presentation. So I think the students here probably already have a questions in mind. So please feel free to ask. You can use the raise hand features and then ask your question, or you also can type your question in the chat box and I will read your questions. So anyone?
Well, it's the afternoon, so probably after lunch. You don't want to, sort of like talk about quite drug topic like this. Having, metabolic disease, obesity, hypertension. But yeah, that's our major concern basically in Malaysia. I don't know about Indonesia, but yeah, in Malaysia we have that problem, and we our people, we don't like to walk. Mm.
Yeah, I think we do have similar things, maybe, but according your data seems Indonesia better, but but still I think we still have the similar problem, the cardiovascular as well as the diabetes still be our major cases, I think. So our lifestyle habit also, I think now it's starting to change. People now maybe more like to do sports. But
Yeah,
still
we are starting to change, but it's a slow progress. And in Malaysia, another thing is the government subsidize the healthcare and medication in a hospital. So the chronic disease is not like a burden like in
Oh,
European countries or in the US, where they have insurance, and you can see the value of the drug in your insurance bill. But in Malaysia, we don't have bills. You go
mm,
to the hospital or any clinic, and you have diabetes. The insulin and everything is dispensed by
cover?
the doctor
Oh,
for you without having, you know, using any documentation like insurance or whatever. You just give your um, KTP, then you
mm,
get the all things for free. So
oh,
it's like of a lavish lifestyle as well,
mm.
and people don't see that this is a burden to our country as well.
I see.
Mm.
But I think similar in our case, but we yeah, for most of the like the chronic disease, it's include in the plan of the government insurance. But still for the drugs, there is some limitation. Like for example, the diabetes for metformin it cover, but maybe for the new medicine like the I forget the name, like a SGL2 inhibitor. It's totally not covered by our government. So still, I think the patients still see that if they have this kind of disease, they know that this insurance will not cover all, like only the general, and for the medicines will be the generic and maybe not so new drugs. So yeah, I don't know. Actually, I don't know that it will be affect the lives, how the patients see the disease.
I think it is good in the sense that the responsibility of, or the awareness of the patient,
Yes.
that if they don't take care of their health and they don't have this healthy lifestyle, it will
Mm,
affect them financially.
yes.
But in Malaysia, because
I
everything
see.
is
Oh,
almost free, even insulin, the insulin
oh,
injection,
oh.
um, even when the patient has already diagnosed with diabetes, is
Mm,
not really their concern until they have CKD, chronic kidney disease,
mm,
or they have um, much more severe complication of
I see.
diabetes. So
See.
yeah, it's good to have everything covered, but that's the consequences on the mentality of
I
the
see.
people,
Yeah,
I
just
think.
yeah. Also, I think it's a new insight for me that it affect how the patient awareness to maintain their healthy lifestyle because they know that it will be covered. And they, I see, I see. Okay.
Okay.
So anyone here that maybe have a question related to Dr. Nomara research, which I think is quite complicated. I'm not sure
It's
I can
not.
follow it well.
It's not. Basically, it's just a combination of, clinical
Yeah.
sample with molecular biology and
Mm.
quite a bit of pharmacology.
Yeah,
But
yeah, yeah.
yeah, if any of the students want
Yes.
to come to our lab, I think
Yes.
you are familiar, Dr. Mehdi, with our lab. You are welcome to hello and come, basically.
Yeah,
So that there's no restriction on that, I think.
yeah, yeah. So yeah, maybe I want to ask first. Hopefully, after these students will then also have a question. So yeah, I'm interesting. When you have the cardiomyocytes from the, I think it's a
Uh,
neonatal. So how
rats.
right? So how old the rats?
It's basically just right after
Yes,
it's
give birth.
born. Yeah,
Okay.
because we don't want much exposure from the environment, especially
Mm,
the bacteria and viruses, because we want the cell to be not contaminated with
mm,
this kind of bacteria. We want to keep, because the cell is primary cell. So
Yeah.
if it's older mice, you have mycoplasma infection, you have other bacteria coming. It is a normal flora in the rats, but it's kind of sort of like, interference for us, especially when we do the metabolomic or when we do the genetic screening, the transcriptomic analysis. So it might give some interference. That's why we use the neonates cell.
I see. So yeah, because I know that doing the primary culture maybe will be challenging, right? So maybe it's too technical, but usually how can you get these primary cells like from maybe mm, one rat, or you need several rats to get the establishment of this primary cardiomyocytes culture?
Um, we actually pull a few babies together.
Oh,
Kind
okay.
of dark to to call them babies, but yeah, a few the neonates bred together, so
I
that
see.
it is faster. But you can actually keep the cell for 2 weeks
Okay.
without much of a difference if you take care of the differentiation, because the cell might differentiate into or become senescent, become all cells, and it's
Mm.
the it's not a problem, but you can see the morphology is quite different, and
Mm.
basically it's cheap because rats produce a lot of babies, right?
Yeah. I see. So
So
you,
you
you
have
need the
adjustment.
special media, right? Special medium to keep it as the cardiomyocytes, or it's just,
Yeah, it's just a normal culture media.
oh, I see. Yeah, because you mentioned that we need to be careful before they can differentiate to other cells. So
Yep.
probably we need to maintain in the specific medium.
Uh, we only keeping it for a few days, so we
Oh.
keep them in very rich serum media, supplemented media with probably TGF beta additional um, enriched hormone.
Mm.
So um, and we have to train the student or research assistant to see the morphology so that
Yes.
the cardiomyocyte does not differentiate into, sort of like fibroblastic like cell, or
Mhm.
but we rarely have that happen.
Change.
Cardiomyocyte is quite easy to get than other types of cell. I think we have because we have one organ, right?
Yes, yes.
Directly excised from the neonates and
Mhm.
just spread on the petri dish, and we just grow it. Unlike thyroid cell, or we used to grow cervical cervical cell. So it's kind of a mixture of um, other types of cell as well. So we have to do the re-isolation of the cell to make sure it's fibroblastic, or to make sure it's just epithelial. But cardomyocyte, you just need to mesh, sort of like mesh the heart of the neonates, and you get the cardomyocyte.
I see. Yeah, it's interesting. So, okay, thank you so much, Dr. Normal. I'm still inviting students here to ask a question.
It's okay. It's all.
Anyone?
It is normal in Malaysia as
Yeah,
well. After the lunch, you become, very
sleepy.
sleepy.
Okay, but Okay, maybe another one more questions for me, Dr. Normal. I'm still hoping there will be students who will be asking. Okay, so regarding the molecules that you use, this PFOS, I'm sorry if I miss your explanation. So this is kind of the dangerous compound produced by the environment, or please correct me if I'm wrong. So what's this PFOS actually? And how in your experiment you use this PFOS? So is it like chemically synthesized? So oh yeah, yeah, what is exactly this PFOS?
PFAS is a plasticizers. It's
Mm,
used in plastic manufacturing plant to
mm,
make the plastic more pliable. So
oh,
if you have any food containers,
I see.
basically you either have PFOS, PFOA, or other types of these plasticizers in the plastic. That's
Mm,
why this compound is really common.
mm,
If not from drinking water, it's coming from your water bottle, or
mm,
it comes from your non-stick pan.
mm,
Okay, if you use non-stick pan, the surface layer of the non-stick pan is covered by PFOS or PFAS or
oh,
any other variant of perfluoroalkyl substances to make it, shiny and to make it withstand the heat. So it's actually available in everywhere. Just we don't
mm,
see it, and it's not like it's famous and people talk about it a lot, but it's a
yeah.
growing concern in Malaysia. Recently, I was invited as well in the Congress of Toxicology in Malaysia, and
Mm,
I think half of the presenters are talking about the impact of this compound in the environment, in water, in soil, in fish, and in
mm.
human as well.
I see, I see. Yeah, okay. Thank you for explanation. So, yeah, I think what your research doing right now is still quiet, still in the very, how can I say, it's a part of basic research, isn't it?
Yes, yes.
Mm,
Um, Although we try to, to do the epidemiological study in
mm,
the population, especially in our area in
mm,
Selangor and in Kuala Lumpur. Um, we try to expand as well the research to the other parts of Malaysia because we have a lot of reports of pollutant, especially in Johor.
mm,
It's in the industrial area in Johor, close to Singapore. So we have collaborators and partners doing sampling in the water sources and soil there as well.
I see. Yeah, yeah, I think it's yeah, quite interesting that because mostly now like in Indonesia, mostly the research like wants to like need to produce something like have a product or something, but I think we know that it's also important to have this kind of basic research. So yeah, if I'm not sure like if I'm also want to do like your similar research, I think I will be so difficult in writing proposals because yeah, mostly they will ask like how exactly it will be implied to the knowledge, so to the, I mean, to the community. So how do you think, Dr. Normala, for yeah, like from this research, what then this research can imply to the, like the community, like what the community should know after knowing these results of your research?
uh, First, as usual, they get scared. That's the report. Basically, actually, Dr. Bedi, I
Mm,
come across this research by accident as well. Previously, I work on the cell model of cytochrome P450, the UGT, the enzyme, metabolic enzyme, phase one, phase two metabolic enzyme. I just study drug interaction, drug herbs interaction, basically. So, but when people ask me, why don't you expand your research to do on environmental pollutant, because it's much more impactful to Malaysia. So yeah, that's when we expand the research. And Malaysia, alhamdulillah, currently Malaysia much more focusing on translational research, not just to get the product, doing research and see the end of the product, but many of the grant nowadays want to see the direct impact to the community and to the country as a whole. Like this research, we measure or basically we survey the level of pollutant in blood sample in our population, because our population is kind of different in terms of the metabolic enzyme. We have different variant of CYP2C9, CYP2B6, UGT1A1. So we tend to metabolize compound quite a bit differently, although Malaysian has Chinese population and Indian population, but the Chinese population, our Malaysian Chinese population is different than the population in China, and Indian population is currently very different than the population in India because of the assimilation with the local community in Malaysia with Malays and interracial marriage between Chinese, Indian, and Malay. So we are unique population, and this kind of research translate directly to the benefit of our local people that probably Western countries or Western researchers will not see this as beneficial as our own people.
Yeah. Okay, thank you, Dr. Normal, for the interesting discussion. So, yeah, I really hope that the research grant not only focus on the end of the the product and the direct impact, but hopefully it will be give a more chance to us as the researcher to do this basic, because even though it's basic, still it will lead to the another research that maybe can have a bigger impact for the society and country. So okay, still I give a last call for every students here. I think it's quite interesting, because at least in our faculty, we still not yet having this kind of topic like from pollutants and check its effect on the metabolic disease. So I think it's quite new for us. So again, last call for everyone here. Maybe you have a questions. Yeah. So if not, maybe I will close this session. So once again, thank you so much for Dr. Norma for the very impressive presentation and discussion. So I really hope it will open the knowledge and give a new insight to all of the students here, our FFUI and other students from another university or country. And if you have any question for Dr. Normala, maybe not here, you can try to contact our secretariat or email, and we can give to Dr. Normala. And if Dr. Normala answer, we can give back to the students for the answer. Okay, so once again, thank you so much, Dr. Normala. So please kind of stay here. We will have a kind of a token of appreciation for you. So, but before I will give back to the MC.
Thank you so much, Dr. Mehdi, and once again, thank you very much to Dr. Normala for your wonderful and insightful lecture. Next, as a form of appreciation, the committee would like to give Dr. Normala and Dr. Mehdi an e-certificate, which will be given by Professor Fadlina virtually.
Aduh, Kak. Buka Reddit ya? Belum balik ya?
Yeah, hello, Dr. Normala Abdul Latif. Yeah, nice to meet you. Thank you for your exciting presentation today.
Thank you for the invitation. I'm very much humble to be given the chance to speak to the students and lecturers of UI. I think I've been to UI very long time ago, in 2016 or 17.
Oh, okay.
Yeah.
Yeah, already long time ago. So maybe you can have a plan to visit us again, maybe in the future.
Yeah, inshallah, if times permit, or if we have further collaboration
Yeah,
next time.
yeah,
I think we are quite close to UI. Dr. Mehdi has been with us for quite some times, and your
yeah.
students
Okay,
have
okay,
visited us as well.
okay. This is our appreciation for your presentation.
Thank you so much. Thank you.
Thank you very much.
Thank you once again, Dr. Normala. For our next token of appreciation, I invite Dr. Mehdi to receive the e-certificate.
Okay. Ms. Mehdi, this is the appreciation for your guide in our evening short course.
Thank you, Prof. Fadlina.
Yeah. Um,
Thank you once again, Dr. Mehdi and Professor Fadlina. Now for our next agenda, we will have a documentation session. So I invite all the participants to turn on your cameras and give your best smile during the photo session. To one of the committees, please guide this photo session.
Hello everyone, please open your camera. Okay, I will start to take a picture. 3, 2, 1. Okay, and next slide. Okay, 3, 2, 1. Okay, thank you.
Thank you for the documentation session. Ladies and gentlemen, once again, I would like to remind all of you to fill in the attendance form and evaluation survey that we have shared in the chat box.
Becoming a pharmacist is my destiny. Being there for people is a must in this job. But it is not that simple. It all started from
Okay, now that we have shared the survey in the chat box, finally, we come to the end of the first half of today's event. May all of us get the value and benefits from the lectures. And once again, thank you very much for your kind attention and participation in this virtual course. Don't forget that tomorrow we have our next virtual course session. So finally, I am closing off today's virtual course session. I am Putri Adrisi signing off, and we'll see you again tomorrow. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Normala.
Thank you, Dr. Kamala.
Thank you, Prof. Thank you, Dr.
becoming
Mary,
a pharmacist is my
We hope to
destiny.
see you again.
Inshallah.
Inshallah.
Being there for people is a must in this job. But it is not that simple. It all started from Faculty of Pharmacy, Universitas Indonesia, the place where I learned